Hi, everyone. My name is Rebecca Gill. I joined the BSGCT board a few months ago as an early career researcher, and today I will be introducing our speaker, Dr. Giulia Massaro. At the end of the session, we will have a Q&A section. You can send your questions on the live chat window on YouTube. To send these questions, you will need to sign to YouTube. And this chat is not private, so please try to include your name and affiliation in your question. And then at the end, we will read all your questions, the speaker will answer them. Alternatively, if you can't log into your YouTube, you can send your question to the email that is on the screen. At the end of the talk, the current president of the BSGCT, Professor Rafael Yanez, will join us for a bit. And without further ado, let me introduce our speaker. Dr. Giulia Massaro is an NHIR Goshby RC Research Fellow in Translation and AAV Technology at the UCL School of Pharmacy. After her master's in research in functional genomics at the University of Trieste and the International Center for Genetic Engineering and Biotechnology, she joined Professor Ahad Rahim's lab at UCL to complete her PhD in gene therapy, working on rare pediatric diseases of infants. Since 2013, she has been involved in many translational gene therapy projects, collaborating with both academia and industry, focusing on rare neurological disorders with unmet medical needs. In 2020, a couple of years ago, Dr. Massaro opened the NeuroGTX viral synthesis facility, a state-of-the-art vector production laboratory for research-grade viral vectors. And today she will be talking us through a step-by-step guide on how to set up and run a research-grade production lab. So thank you and welcome, Giulia.
Thank you very much. Thank you for the introduction and thank you for inviting me. So as said by Rebecca, today I'm going to talk to you about how we started this VectorCo. This is a research academic VectorCo facility, and I'm going to talk to you about how we came about with the idea, what funding is needed, and how to set up in general and manage a vector production lab. So the NeuroGTX facility has opened officially in November 2020, although the initial funding was already available from before, from April 2020, I think. But then, of course, it took a long time to set up everything. So officially we started in November 2020. The main funding body for this facility is the NIHR Great Ormond Street Hospital Biomedical Research Center, who funded part of the initial equipment needed for the facility and is actually paying my salary. And then we had also an additional support from the UCL School of Pharmacy. So Professor Stephanie George at the time was the head of the department, the pharmacology department at the School of Pharmacy, and she was successful in her grant application for Capital Equipment Fund, UCL, and with that money, she contributed to buy some of the equipment needed to get it started. So I just want to spend a couple of minutes on the concept of the facility. This is not just a place where you contact us, you pay, and someone ships a little vial with the viral prep in it, and you don't have to do anything. It's completely the opposite. It's a very teaching based platform. Is a very much academic concept, and which this I mean that when you contact us and we decide to work together, you actually come to the lab and you yourself are making the prep. Of course, I'm there, I'm here to help, to guide, to suggest, but the person, the user, is the person coming and learning how to produce these products. So the scope of the facility is to give access to a production laboratory for research grade viral vectors. And the remit is to design and produce these vectors, and overall to accelerate preclinical studies. So then these studies and this research can move towards the clinic faster. So myself and the UCL team behind the facility have very broad and a long expertise in gene therapy. And myself, I've been working for the last 8 years in translational gene therapies, and the applicants, the various Professor Waddington, Kisson and Rahim, they have decades of experience in gene therapy. So we have a strong team behind us. So as I said, the facility provides not just a place and the equipment, but also a guide, and we advise researchers and users. And we have a very, very varied user base. We have people that they're not in gene therapy, they are not in the gene therapy field, they don't know anything about gene therapy. They might just need some GFP for a reported gene study, and let's say, and they're not moving towards translation into the clinic. We have some users that they're experienced users, they're already working in gene therapy, and for whichever reason they might not have access to a lab, something broke down, or is just busy and they need a prep made. So we have quite a varied use base. And according to the person that is contact me, I give different levels of help and advice. So if a user that is not in the gene therapy field and never design or use a viral vector, contact me. I'm more than happy to help them from the very beginning, from the plasmid design. How do I acquire an expression plasmid or capsid plasmids? How do I design them? Where do I get the DNA sequences from? And all the way down to the downstream application, for example, like in vivo application. How do I inject these preps, these vectors into animal? What's the dosage, and things like this. So as I said, it's quite a varied user base. And the whole scope is provide to teach and provide opportunity for various kind of research and join the lab and use the facility. So in terms of equipment, we start from this picture. It looks big, but I can tell you that is definitely not big. This is the very first room that we started with. We had some of the equipment made available by the department through the CEF funding application. So this was around £100,000, and with this we bought a tissue culture cabinet, a multi-centrifuge, a high-speed centrifuge, various small bits of equipment, and then we had, of course, made the actual room, the actual lab, GMP compliant with all the health and safety addition. And then with some extra money that was left from the BSc calls, around £35,000, we bought other bits of equipment, an HPLC machine, scale, water bath, fridges and freezers, pipettes, everything that is needed for tissue culture, a small microscope, some plasticware, and a couple of rounds of consumables and chemicals for the first few preps. So I'm sure I don't have to go into detail about vectors for this audience, but I just want you to introduce what platform we're using in the facility. So at the moment, we're using adherent HEC293 cells, and we use the standard triple transfection method. This means you have your 3 plasmid with the gene of interest, your wrap and cap I'm talking about AAV mainly, and your helper genes. You transfect the cells in dishes or in flasks. The cells are packaging the vector, and then you collect them, purify, concentrate, and at the end you check for titer and quality of the prep. So at the moment, we are using PI Max as transfecting agent, but I am testing different products in the market and check if different products has different effects on the yield and quality of the vectors. In terms of purification, we use mainly two methods. We have available an HPLC machine, as I previously said, and an ultracentrifuge. So for the HPLC, I'm using these columns, the porous AVX columns from Thermo Fisher, and this is because, of course, every user wants to produce a different capsid, a different vector. AV vector. So I couldn't afford at the beginning to have loads of different columns for each serotypes, and these AVX columns, they work quite well with different AV serotypes. And as you know, HPLC purification, so affinity chromatography, is quick and easy. It's a nice method of purification, but of course you can't distinguish between full capsid and empty capsid. So it's something that I point out to users. Some users prefer to use HPLC, some they go down the route of ultracentrifugation. And I have to say, despite being longer and more painful, ultracentrifugation is still my favorite methods. So at the moment, I mainly use density gradients with iodixanol, but of course we have the centrifuge, we have the tubes. So if needed, if some user prefers to use cesium chloride methods of purification, we can still do that as well. So although I've been making virus for the last 8 years, way before the facility opened, and I had a very well-established protocol, literally 2 doors down the lab in Professor Rahim's research group lab. Just the fact that we move from one tissue culture to the another, one centrifuge to the another, using the 17 mLs Beckman tube instead of the 14 ml Beckman tubes, all of this changes the protocol. So before having the first user come in and produce their vector, I had to run multiple test runs to optimize the new protocols according to the new equipments that we had. So this means that for the first few months, I was making loads of preps, and I was comparing different methods of transfection, different methods of purifications to find the optimal protocol for this new room, for this facility. In terms of quality controls, QC offer at the end. At the moment, we offer vector genome concentration via qPCR. And of course, as you know, it's an easy, quick method. It's quite reliable for the quality of preps that we're producing. So it's research grade vector. So this is usually enough for our user to have an idea of the concentration and how to calculate the dosage when they go into the in vivo studies. Oh, sorry. Back. So then we can also run an alkaline gel to check for the quality of the genome of the vector. And this is not something that I usually do routinely, but I do when I have difficult products. And with this, I mean when, for example, the genome is extra size, is over packaging. So I want to check the percentage, how many of these viral particles have the full length genome, and how many might have chopped version and shortened version of the genome. And then we can run the STS protein gel to check for purity of the prep and the presence of any contaminants. And you can see here, this is a very nice gel, nice and clean. And finally, we can test the endotoxin level of the protein. And this is, again, something that the users sometimes ask, not always, when they have slightly be more sensitive and let's call it precious experiments. So they can ask for this. So the expected yield for an AAV prep is around 10 to the 13 VG per ml. For lentiviruses is around 10 to the 8. But then, of course, as you know, every prep is different, every gene of interest is different, every capsid or envelope is different. So this is just an estimate. Some genes can be a bit toxic for cells. So when you transfect your cells, they can cause cytotoxicity. Some capsid or some envelopes can produce better than others. So all of this reflects on the final yield, the final concentration. So some preps may result with a lower titer, but again, I always make this very clear to the person, to the user at the very beginning of the experiments. And then obviously COVID hit, and I'm sure that all of us had struggled a lot in this last couple of years because of the pandemic. And of course, this had a big impact on the facility because first of all, we couldn't get the equipment in because of the restriction. So moving, getting the new equipment in, getting the engineer to come and install all the equipment was a very long process, and that's why it took us a very long time, many months, to start actually producing some vectors since the opening. And then at the same time of the pandemic, UCL was going through a reshuffling. So the GM safety committee was all around the world, and it was difficult to get the hold on them. So it took me over 4 months to just get my risk assessment sign off. So until that wasn't in place, I couldn't do literally anything. And then until July 2021, the school had quite a tough and restricted access to the school, to the building and the facility. And that means that only one person could stay in the room because it was a quite small room. So it was a single person occupancy. So while the person was sitting in the hood doing the splitting the cells, I was standing in the corridor shouting how to split the cells, what to do, what not to do. And then they would come out, I would go in, check if everything was fine, and then we would swap place around. So it was quite convoluted, complicated process. And then, of course, all the reagents, although with the first batch of the first pot of money, we managed to get some consumable for a couple of rounds of preps. It was very difficult to get a hold on consumable and plasticware. So this further delayed the operation in the facility. And then last but not least, the tissue culture cabinet broke, and I was for almost 5 months without tissue culture hoods until August 2021. So my line manager, Professor Hadrian, very kindly offered the use of one of his hoods, but then of course I had to coordinate with my research group and the user from the facility. So again, it wasn't easy, but we managed, and now we're fully up and running. In terms of user base, I pull out these numbers from the launch webinar that UCL Translational Office offered in August 2020 to promote the facility. So mainly people were interested, as you can see here, in AAV gene therapy. the vast majority was working in the field of neurology, neurobiology, and most of them, more than half of them, they were already working in gene therapy. But many of them, they were not in the field, and they were interested in getting into the gene therapy field. So that was quite promising and good news for us. So from the opening, November 2020. We got more than 20 users so far. They were mainly from UCL, but also I had people from other institutions, although they were not, they were somehow connected to UCL via a secondary supervisor or a collaborator. So they heard about us via the collaborator at UCL. And most of the people, I would say 90% of the preps, they were AAV preps so far, and then we had some users asking for some lentiviral preps. In terms of projects, I'm very pleased to say that most of the projects that we've been involved in, they are moving towards the clinic and towards along the translational pathway, and our help, using the facility and getting these products done, help many researchers with the funding applications. And many colleagues, they were successful in application for example, the LifeArc Philanthropic Fund, the Wellcome Discovery Grant. Some people, they're filing IP on the plasmid and the vector that we designed together and we produced together. So it's very good news for us. In terms of the research field, as expected, most of the users, they come from the neuroscience field. And this reflects on the AV, mainly capsids produced. So 70% of the capsid that we produce so far, they are AV9, PHPB, PHPEB. But we have some other research fields like nephrology and other fields, and of course other capsid. And because the main application is neuroscience, the main research field is neuroscience. The most used application is for, of course, in vivo studies. But we have people contacting us also to develop in vitro analysis. So the facility has been very successful, and the income of the first year and a half has been invested in reagents, new equipment, and we decided to expand the facility, not just in scope and capability, but physically. So we moved one door down in a very nicer, bigger, larger lab with windows. And this allows us to accommodate a wider use space. We can buy new equipment and have just more space to work in a nicer environment. And this led us also to start some conversation with UCL and to designate the facility as a science technology platform, so something that can be available to a wider user base among UCL. The facility is not just a place where you come and make vector, is also has also research concept, is an academic concept, and in this sense we work also towards new AAVs, especially AAV technologies. And to do so, we started some collaborations, and we have a very close and successful ongoing collaboration with the UCL Biochemical Engineering Department and Paul Biotech Center of Excellence. This is Luis here, is my PhD student. I just started a few months ago, in collaboration with Dr. Kazim Rafiq at Bioengineering, and he will be working on AV technology and capsid design. As we all know, everyone is looking for the best capsid for the application, and Luis is going to look into this, touching upon different different ways on how to engineer this capsid. So there is also research going on in these facilities, not just a service that we provide to external users. In terms of the starting material requirements, so what I ask when to people when they contact me, so they contact me and we have a call to understand what is their needs, what stage of the project they are. And then I send them my requirements for the starting material. So the users are required to bring their own, obviously the gene of interest plasmids, and also the capsids in the AAV capsid plasmid. So in terms of the gene of interest plasmid, of course, the genome size needs to be checked. These plasmids, they need to be produced in endonuclease and recombinant deficient cells, prepped in with endotoxin-free kits, and they need to be checked, possibly sequenced. And I asked to do a restriction cut with the SMAR1 enzyme to check for the ITR integrities. And these are the scale of DNA that I ask to bring over for a prep. And same goes for the capsids, AAV capsid, and if we're talking about lentivirus, for the gene of interest lentiviral plasmid. Services and costs. So by default, we offer two production scale, a small scale. There's about 20 dishes of cells to produce 300, 500 microliter of concentrated vector. And a larger scale is about double, 40 dishes for around 800, 900, 1 ml of concentrated virus. But we're very flexible, and at the moment I've done all the way from 60 dishes for large preps to 5-10 dishes for small trial preps, if someone wants to just give it a go and see how it goes. So we're very flexible, and we try to accommodate everyone's needs. In terms of costs for AAV, we ask something between 1,600 and 2,000 pounds. For lentivirus, a bit less, between 600 and 1,000 pounds. But then again, we can be flexible. I had early career researcher, PhD students that got, for example, I don't know, £1,000 little grants to spend in vector production, and they don't have extra consumables. So we can try to be, again, accommodating. Or some people, they come and say, look, we want to make 10 preps. Can we have little discounts? So we can talk about that, as long as we cover the cost. So this facility is run at cost. We don't make any money. I certainly don't make any money out of it. And what we ask is purely for the consumable for the next prep, and to put some money into the pot for the service of the various pieces of equipment. So the user can decide if they want to pay an extra bit for insurance in case the prep doesn't work and fails. So we can make another prep without additional costs. Although, again, I have to stress the fact that I'm very accommodating. So even if it's something happen or at the very last minute you can come, I'll try to use all the equipment and the cells for something else. So at the moment, I never been in a difficult position to ask for extra money to the users. So the business model, as I said, is running at cost. So we're not making extra money. So everything that we, all the money that we get is immediately invested in new consumables, survey contracts, and buying new equipment. The payment method at the moment, so the fees are transferred via interdepartmental transfer, and that's why for now we keeping it close to only UCL users, because having people from other institution would make this business payment method a bit more difficult, and they would have to pay extra for extra overheads and all these things. So at the moment, we try to keep it for UCL users. But I went around this a couple of times. I had people from other institutions contact me and wanted to use the facility. And what I did, I started an actual collaboration more than asking for some money. And so we got around there, and they managed to come and use the facility, learn how to make some vectors, and we got a bit around the problem of the payment. So when the user contact me and we decide we have a strategy and we have a plan, I send them a quote. If they're happy with that, I send them down an agreement form in which there are stated all the user's info and contacts, the description of the work that is going to be carried out, the Plasmid requirements, so they have everything written, everything there at the availability. And then I ask if and make sure they have an MTA in place for the use of these plasmid or vectors. We all know this is quite a crucial aspect of what we do. And if they don't, I help them sort this out. I tell them where they can buy, for example, the plasmids, and sort out this agreement before starting the work. And also I have to make sure that they have a GM risk assessment in place to use these vectors in the lab. And then finally, there are some terms and conditions regarding, for example, cancellation. As I said, I don't want to ask for more money to people. I try to always be accommodating. So if you have to cancel and I can use the cells for another purpose, something else, I'm not going to ask any extra money. Unless you literally cancel on the morning and I can do anything. I lost a lot of time, my time, and money preparing all the cells for you. And then, of course, I can't take any responsibility for the failure that is to negligence. So I give the protocol. I'm with the users all the time. I'm always available to show, to help out. But then if someone is not following my advice and gets a massive contamination on the cells, I really can't do anything about that. And I can't take responsibility for that. So this is something that I make very clear at the beginning when the users come to me. And then, of course, the quotes are non-negotiable once the work has started. In terms of future horizon, well, I have to say that we've been doing quite well in these last couple of years, and the BSc has put forward a new funding application for 2022 for the 5 years, I think, and part of this money is going to go into supporting, again, the facility. And as future perspective, we are thinking of recruiting a research assistant, an experienced research assistant. At the moment is me, myself, and I, and I'm doing everything, and I'm spending a lot of time because it's very time consuming. And as I said, I'm there with the people all the time for 2 weeks. So it's very, very time consuming, and I have to do this on top of my actual research work. So having the help of a research assistant, a technician, or a scientist, that would be obviously beneficial for everyone. And we can, with that, also expand the user base. And then, as I mentioned before, we would like to designate the facility as a science technology platform. And then I have to say I'm very pleased. This is news of a couple of days ago. We've been successful in this grant, the Capital Equipment Fund, the CEF4 call. We got awarded around £250,000. And then on top of that, we're going to have another additional support from the UCL School of Pharmacy of an extra around 30K. And this money is going to be used to buy a digital droplet PCR. As we all know, the yield and the titer of vectors is a very hot topic, and the best way to to quantify this is via digital droplet PCR. So this machine is going to be used for vector quantification, yield quantification, but also it can be used for downstream applications. So once you produce your vectors, you inject your animals, you can do biodistribution studies and other application. And this can be, will be open to use for from other UCL users, not just people that are coming in the facility to produce the vectors. And then the next bit that we want to expand on is moving to suspension cell lines. So as I said, at the moment, we're working with transextraction on adherent cells. It's a system that is established, it works well, it's fairly easy, and you know, it's okay for the needs of the users at the moment. But we all know that with the demand of high titers and large batches, adherent cells is not a platform that is sustainable in the clinic. And all the companies and GMP manufacturer, they already moved to, of course, suspension cells. And this is something that we want to do in the facility as well. And so the aim is to move to suspension cells using serum-free and antibiotic-free media to reduce contaminants and, of course, reduce the cost, reduce the footprints, and being able to scale up the production and accommodate at the same time different users. So overall, I have to say this was started as an experiment, but it turned out to be a very successful enterprise. In the last 2 years, we've been financially self-sustained. We expanded based on this initial success, and we are now able to accommodate a wider user base. And at the moment, I'm fully booked week after week until the end of Q2 of 2022. And I keep having requests from people wanting to use the facility. So this is very good news for us. We haven't had a failed prep so far. We just had one very low titer prep, but we're figuring out, and it's not something about the production protocol, but something about the upstream plasmid design. So I'm working with people at different levels. And I have to say that many users, they actually came back to use the facility again. So that was, again, a good sign for us. And as I said, we produce a lot of a variety of vectors and capsids very successfully. And as I said, the concept of this facility is a teaching platform and a research platform. So we're always seeking to improve manufacturing methods and scalability and AV technology in particular. So I want to conclude with some challenges. The main one is, why I didn't get high titer or a titer. I was expecting this number and I got this other number. What happened? So it's obviously there is a lot of customer service and managing the expectations. So I always try to be clear at the beginning, especially for people that are not into the gene therapy field, so they've never done this before and they might not understand how the biology works. So we can go through the process and then something happens, not because of the users, but because of the actual biology, and the titer might not be as good as you thought. So how you manage the expectation and how you prepare users for every possibility. And of course, there is a balance between teaching the person, so giving the protocol, teaching them, make them understand how the biology work, how the process work, how the manufacturing process work, and let them do the work. But at the same time, it needs to be made at high standards, because otherwise it's a waste of time, my time, their time, a waste of money. So it's really not worth. So it's difficult to find sometimes the balance between the two. And you might think, Oh God, if I could make it myself, it'll be better and quicker and get it done and get over and get on with our lives. But of course I can. So I need to accommodate everyone's need, and this requires a bit of, as I said, customer service. And in terms of the facility itself, is the big question is, of course, how long can this model last? So as I said, at the moment, the facility is doing very well, it's self-sustaining. We had a nice pot of money that we're investing in new in buying new equipments. But how long this is going to last? So our prediction are that hopefully for the next couple of years, so within the first 4 years of life of this facility, we still will be in the same trajectory, in a stable trajectory, and we will be able to again cover all the costs. But then, of course, we have to think about what if the user base drops for whichever reason, what we're going to do, how we can sustain the expenses. And here comes in place the more research technology part of the facility. So can we provide new services? Can we provide new capsid, for example, and compensate for a lower user base with something like this? But then at the same time, it's difficult to gauge if the users will be willing to pay maybe a bit more for new technologies. Will they be willing to pay a bit more for new novel capsids? Do they have 150 to 100 pounds extra to run a ddPCR titration instead of just a normal PCR? These are difficult questions. I don't have an answer yet. But as I said, hopefully in the next few years, the trajectory is going to stay stable and we're still going to be able to offer this service. So to conclude, this is the website that we have. We have a small webpage in the UCL Therapeutic Innovation Network cluster, and these are contacts. If you have any questions or if you're interested in work with us, please feel free to just drop us an email. And finally, of course, I want to thank everyone that contributed to the birth, the genesis of this facility, the BSc, School of Pharmacy, and everyone involved. And of course, the BSGCT for inviting me and talked to you about my last couple of years. So with this, I leave you with the last slide.
Thank you, Giulia. That was a great talk. I'll let Raphael say a few words before we move on to the Q&A section.
Thank you, Rebecca. First of all, I want to thank Giulia for an excellent presentation, which obviously is a little different from our standard research seminar. So extra effort for Giulia to prepare for this. And that's very much appreciated. Giulia has been elected to the board of the British Society for Gene and Cell Therapy recently, and it's a pleasure to have her as a representative of the board here. And the seminar itself has been really interesting. Giulia, I think obviously AAV is really fashionable technology, highly effective, with a number of marketed treatments already, and so many more in the pipeline. And it's great to see that an academic institution like UCL has been able to set up an effective platform in order to make this available, not just necessarily to the gene therapy community, but perhaps other people who have an interest in using these vectors, but not necessarily the capability to produce them. So that has been great. I think that Rebecca was going to introduce our next seminar in this series, and then we can crack on with questions for Giulia. Is that so, Rebecca?
Yep, thank you. So before moving on to the Q&A section, I would just like to invite everyone to join us in our next online event, which will be the 16th of March. This seminar was organized by the Early Career Researchers Subcommittee, and we're going to have Dr. Angie Miller from LifeArc as our speaker, and we're going to try to help early career researchers to solve the $1 million question: a profession in academia or in industry, is there a middle ground? So please join us if you can. Angie's a great speaker, so I'm sure you'll enjoy it. And I would also like to invite everyone to become a BSGCT member if you're not already. Some of the membership benefits are reduced rates for our conferences and meetings, free membership to ESGCT for the 2022 Annual Congress, and board and subcommittee opportunities.
That's great. Thank you, Rebecca. So if I may kick off with a first question, Giulia.
Yeah, so I'll stop sharing. So now everyone should see us.
Yes, I think that's correct. So Giulia, on the back of that slide, can I ask, your career at the moment seems to be a little bit halfway between academia and providing a service. So
yes.
What attracted you to following this pathway, and what advantages and disadvantages can you see of it?
That's a very good question that I ask myself every day. So as you say, it's perfectly said. I'm a bit in between. So I was attracted to the idea because, of course, this is what I do. I can do well, and I like to deal with people. I like to teach people, and this also gave me the opportunity to start many collaboration, not just for the facility, but also for my for my own personal research career. It's very difficult to balance the two because, as I said, this takes a big chunk of my time every day, every week. So I have to balance the two, and sometimes it's difficult. But again, I want to accommodate and be available for everyone. So I try to give the priority to the people coming for the facility because I know that maybe they're struggling, they have a deadline. So I try to give them as much as I can. And then I stay a long time in the lab, and in the evening after 5 p.m. I start doing my actual work. So it's a bit tough, but I'm enjoying it. But that's why when I presented the future perspective, the next round of application would probably include recruiting a research assistant or a scientist to work alongside me to take over the everyday aspects of the facility, while I can stay a bit more on the managing side. I can still help designing the studies, designing the plasmid. I don't mind that. But thawing cells, and I expand the cells, I split them, I prepare everything for them until they arrive on Monday morning. So it's quite time consuming.
Absolutely. Because obviously when you say that after 5 p.m. you do your work, you mean actually you carry out
my own research, yes. Yes.
Right.
So it's been a few tough months, but as I said, I like the job, so I'm happy to do it. But if I can get some help, that would be much appreciated.
I can understand that. And Giulia, when you say that the facility has covered its costs with these relatively modest charges for the prep, is, just so I understand, is it up to a point that the costs have been covered? Because obviously the facility is supported by a number of research grants and internal funding from UCL. So those costs are in addition to the income received from users.
So the initial funds was to buy the initial equipment, so the money is gone, and then is to pay my salary. So everything that we get from the users gets let's say that all the equipment has been, all the service charges and engineer calling and all these things is perfectly covered by the money get from the users, and all the consumables and cells, plasticware, and everything is covered by that. And I have to say, because we've been quite successful, I have a nice pot of money. I have an extra bit that I'm now reinvesting. I'm buying 2 new incubators and things like that. Because I said we had more than 20 users, but each users, they came and asked for more than one prep. So really, for each person, we could get up to £10,000. So that's what I'm saying. We've been quite successful. It's been quite a good model so far. And because we don't want to make money out of it, we want to immediately reinvest all the income that we get in this, new equipment, service charges. Now with this DD-PCR that is going to come, the service charges are like 20k per year, something like that. So we'll have to design a very good business model to cover that.
All right. Thanks very much, Giulia. Not to hog the conversation, Rebecca, do we have some questions from the chat?
Yeah, we have a few questions from our audience. So let's start with that. So Robin Bell from Imperial asks, do you have any strategies to help minimize the negative effects of toxic large transgenes on vector titer?
Again, $1 million question. That's why, let's say I have an initial consultation with the users. And as I said, if it's someone that is not in the gene therapy field, I try to make them design the plasmid and the vector in the most neat way possible. I help them finding the right vector for their application. Sometimes I had people come in with the huge transgene and they wanted to prepare it anyway. I said, look, I told them the titer is going to go down because that's what happened, especially for AV. As soon as you go after the 4.95, 5.1 kb, the titer goes down. And honestly, I haven't found a way to get around this problem yet. There are a few things that you can do upstream during your plasmid design phase. If you can split, I don't know, let's say it's channel or something like that. You can split it in 2 parts and co-administer, or just having some sort of strategies upstream. But during the production, actual manufacture methods, I don't have an actual answer for that. I'm sorry.
That's okay. Thank you, Giulia. The next question is from Jack Hickmott from Imperial. What was the process for getting capsid plasmids from other institutes? Where did you get them from? And was there any pushback around using them to provide a service?
Yes, so we get a bit around that because we're not a company, we're still a research institution, so we can get around that. And the final user is still a preclinical research a researcher, academic researcher, so we can get around that with that. The problem with the MTAs is that, of course, the MTAs should be held by the final users, that they're actually using the viral particle. So it's not on the plasma, it's on the viral particle most of the time. So we can produce it fine at School of Pharmacy, but then the person in the next lab, in the lab where the user is, and is going to inject this capsid and is going to write a paper about it or present it, they need to acknowledge and they need to have a contract in place with the original lab. So the easiest way for us, we found out, is basically that the user, if they don't have it already, they buy the plasmid capsid from website and companies like Adgin, which provides already and sort out the MTA with the provider already. So everything is done. UCL deals and liaise with Adgin and the third party, the provider, and everything is done via that. So everyone is covered, no one is suing us, and everyone is happy.
Yeah, no lawyers getting involved.
Exactly. But again, if you're not in the field, you might not know these things, you might not think about these things. So that's something that I make sure every time at the beginning, so then everyone is safe.
Okay, thank you. Next question is from Riccardo Privolizzi. Fantastic talk, fantastic service. Do you have experience or envisage the development of rapid protocols for initial screening of multiple AAV vectors rather than half of full or full-size preps?
Yeah, so that's what I was saying about when I was talking about the different scales. I've been doing 5 dishes, like very, very small scale preps, and just to check if everything works fine, if it's a transgene that you never produce or a capsid that you never produce. So we can do that and have a quick screening. If then you're talking more about how do you actually screen these products in the downstream, in the manufacturing, in the production part, then, well, that is something that my PhD Lewis is looking into, because he's going to create all these massive libraries of different capsid, and he will have to find a smart way to test all these. So yeah, he has 4 years.
Yeah, that's very interesting. Thank you. Next question is from Carly Bliss from Cardiff University. Congratulations on setting up such an impressive core facility. Do you think the service will eventually expand beyond UCL users?
Hopefully, yes. The advantage here is that we all know how expensive these preps are when you buy them from a company. And we also know that not everyone has availability to produce this thing. So this is a service that could, be beneficial for a lot of other people. And hopefully, yes, my aim is to expand to other institutions, but there is a lot of paperwork that has to be done before.
Yeah. On that note, I do have a question, Giulia. Do you think that, congratulations on expanding and getting those nice windows.
Yes,
in the new
I can see the sunset.
That's always a good thing. I was wondering if when you change to a bioreactor for the cell culture, do you think that's going to affect or improve the cost of the batch?
So we're not going to start directly with the bioreactor. I think we're going to start with flasks, shaking flasks, just to get the new protocol going. Bioreactors are very expensive, so I don't think we have the capability yet. But I have to say that the collaboration with the Department of Bioengineer is quite useful in that sense, because that they have a very, very nice setup. They have bioreactors. Lewis is going to be is an adherent bioreactor. The iCell is nano. But nonetheless, they have also suspension cells bioreactors, mainly for lentivirus production. So if it's not directly us, we are in a good position with our collaborators to possibly move to bioreactors. So, yeah.
Mm-hmm. Perfect. We have a question from Nora Clark from Imperial. Do you offer access to a plasmid database store, especially those new to gene therapy, or do you require everyone to provide all of their own production plasmids and genome plasmids?
So go.
It might be interesting to pair with cloning services in the future, maybe.
Yeah, absolutely. The problem there is, again, who holds the IP of these things? If I design a plasmid, I have my own plasmids, and I spend hours designing my super duper plasmids. which I don't mind share if it's a collaboration, but I can't just give away an IP, an intellectual property that can be mine or someone else's without any sort of contract in place. And this would make the whole concept way more difficult. It would be amazing. Obviously, it would be amazing. But I don't think we're there yet. Of course, having in pair, as you were suggesting, a cloning service facility, that would be ideal, but again, we're not there yet. The other idea was, as future perspective, is to just produce some off-shelf products, maybe some, the classic GFP, RFP, some of those preps that can be used by different people with plasmid that they don't come from other sources that I design, and I decide, okay, you can have the IP for free. You just pay for the vector already done, and you don't have to come. So that is something that we're looking into, to just provide some off-shelf products.
Okay, I think we're gonna squeeze one last question before we wrap things up. Robin Bell from Imperial asks, have you explored many different lenti pseudotypes or just VSVG so far?
As I said, I don't do many lentis. I'm an AV person, they're my favorite. I made some lentis with some people that they came to the facility. At the moment, we just have the lenti plasmid. But then again, if someone comes and has their own different plasmids, or they want to produce something different, I'm sure we can work it out. But I have to say, my actual expertise and my research is in AV vectorology and technology. So I personally don't have the capability of doing this work for lenti plasmid as well. But yeah, again, I'm happy to. I like to do science, so I'm happy to do research with other collaborators.
Perfect. I think that's all from the Q&A section. Thank you so much, Giulia, for this amazing talk. I think everyone working in gene therapy is obviously interested in this topic, so I really enjoyed this talk. Also, thank you, Rafael, for joining us, and thank you to everyone that is here with us. And we'll hope to see you on our next events as well.
Thank you very much.